Before we begin, I'd like to acknowledge the traditional custodians of the lands on which we gather today, the Bidjigal people of the Eora Nation, and pay our respects to their elders past, present, and emerging. So for today, I guess a bit of housekeeping. If you have questions in the room, please use the microphone on the right-hand side here. And if you have questions online, please put it on the chat box. And so for today's seminar, I have the pleasure of introducing Assistant Professor Michael Peluso, who's visiting us from San Fran. Mike was here at the Long COVID workshop and conference in Melbourne last week. So we thought it's a great opportunity for him to present, his amazing work in the long COVID space, as well as his trials in HIV cure. So please, I'll hand it over to Mike.
Hey, everybody. Thanks for being here. I'd also like to acknowledge the Bidjigal people of the Eora Nation and pay my respects to elders past, present, and emerging. It's really a pleasure to be here with you. It's my second time to Australia, but my first time to Sydney, and I've enjoyed it so far. I thought what I'd do today is provide an overview on what we're working on at UCSF in terms of both our HIV cure program and our long COVID program. And these have actually become highly synergistic and kind of intertwined over the years, which has been really fun. And I think, quite enjoyed how the two programs kind of inform one another. I want to begin by acknowledging our research team. I was telling Chan before that at the beginning of all of this, our team was about 5 people. Now it's about 30 people working across both HIV cure and long COVID. And it's really probably the best part of my job is getting to work with these people every day. So a little bit about me. I'm from New York originally. I grew up in the suburbs of New York City, went to public school there. I did my undergrad at Columbia in New York City in neuroscience and evolutionary biology, and then spent a year in the UK at Cambridge doing some training in public health. And then I went to medical school at Yale, and instead of a 4-year medical school program, which is typical in the US, I did a 5-year program and had sort of a dedicated research year, where I worked with Serena Spudic, who's an HIV neurologist. And she was sort of my first research mentor and really got me interested in HIV. Then I went up to Boston and did residency in internal medicine and global health at the Brigham, and moved over to San Francisco to the West Coast in 2017 to do my infectious disease fellowship. And I went to UCSF really to work with the HIV cure group, in particular Steve Deeks and Tim Henrich, with the goal of learning how to do sort of experimental medicine HIV cure trials. My overall focus is really on trying to address difficult problems in ID by working on clinical translational science. So I'm not a laboratory scientist. I'm really based in the clinic. And what I do is try to identify important clinical problems or interesting clinical phenotypes that I then partner with our laboratory collaborators to try to answer in the lab. And then at the same time, work on identifying findings in the lab that can be translated back into the clinic in the context of the experimental medicine trials that I mentioned before. So my initial intention had been to do all of this within the realm of HIV. But then, like most people, COVID sort of threw a curveball, and now I spend pretty much an equal amount of time on both HIV and SARS-CoV-2. So I thought I'd start by providing some updates on our recent clinical trials in the HIV cure program. So a lot of my focus in HIV has been on models of HIV control off of ART. So my initial fellowship work was focused on mechanisms of spontaneous HIV control. And so that can exist on a spectrum between viremic control, which are people who are able to maintain viral loads in the 1,000 to kind of 5,000 range despite not being on HIV meds. Then we started to do a deeper dive into one extreme of that spectrum, which is elite control, which basically means that people can effectively suppress their virus to levels that are undetectable on modern assays, despite not being on any medications. But most elite controllers will eventually lose control and go on to progress. And so there's been a lot of interest recently in characterizing these models of what's called exceptional control, which are basically elite controllers who maintain control for 20 or 30 years. There are only a handful of people who have been followed for long enough to demonstrate that they have achieved this. And that was a big focus of my fellowship. Over the last few years, though, we've really shifted our program to understand models of what we refer to as induced HIV control. So that could be post-treatment controllers, people who, are on HIV meds and stop them for one reason or another, planned or unplanned, and are able to control their virus after they go off of meds. And then post-intervention control, where we basically try to induce one of those phenotypes in people by giving them a variety of immunotherapies. So when we're thinking about post-treatment and post-intervention control, we're really thinking about multiple parameters that can occur kind of virologically that may correlate with control. So what we're looking at here is, plasma RNA level on the y-axis and time on the x-axis. And, people are on ART and they're obviously suppressed. And then when people stop ART, we look at a number of different parameters within the rebound. So the first is the time it takes for the virus to reemerge, time to rebound. The second is the sort of explosiveness with which it reemerges when it does come back, which is typically exponential. When the virus comes back, it comes back quickly. It often goes to very high levels. So we pay attention to kind of the viral load peak that people achieve when the virus reemerges, and then ultimately what their viral load set point turns out to be. And the goal of all of the different immunotherapy studies that we do is to really try to drive down that set point from what is typical, a high level in the 10,000, 50,000, 100,000 or higher, to lower levels that are in the range of the viremic or elite controllers that I mentioned previously. So most studies of post-intervention control and post-treatment control have really been focused on studying people after they've kind of already reached the set point to understand how they may have gotten there. Our group is really interested in this very early period that occurs when the virus is sort of first reemerging and encountering the immune system again for the first time off of ART. And we refer to that as the intercept. And so we think that the immunology of the intercept is really important in figuring out who is destined to go on and control. Studying this time point in people is like exceptionally difficult. It's difficult for a variety of reasons. It's unpredictable when a specific individual will rebound. It can take 2 days, it could take 2 weeks, it could take 2 months. So capturing that exact moment can be really hard. It's based on a variety of factors, some of which we can anticipate, but most of which we probably can't anticipate. Once rebound starts, it's exponential. And so we've likely already missed the window to capture those sort of key biospecimens that would inform the biology. A lot of the interesting biology is probably taking place in the tissues. So if you wait to see somebody's virus reemerge in blood and then schedule a tissue collection procedure, now you're sort of way out of the window. And so most studies so far are not designed to really capture this key period because it's very logistically challenging. So the way that we've addressed this in San Francisco is on a grant funded by Lily Cohn, shown here. We basically designed a study that was built around maximizing the likelihood that we would capture the key biological time points. So what we did in this study was we took people who were known to be either non-controllers or controllers before they had ever started ART. Everyone in San Francisco is pretty much on ART, regardless of their phenotype, because that's standard of care. And then what we did was we did a number of baseline measurements while they were still on meds, including leukapheresis, serial blood measurements, gut biopsies, lymph node FNAs. And then in a very, very coordinated manner, we interrupted their ART and basically saw them 3 times per week for up to a month, so that we would be able to retroactively identify the moment at which the virus came back, even though we couldn't get that result in real time. In everybody, we repeated those tissue procedures between 1 and 2 weeks to kind of capture what was going on in the tissues during those earliest phases. So far in the study, we've enrolled 20 people, including 13 non-controllers and 7 prior controllers. And we're now doing a deep dive to really understand the biology. What's happened clinically is fascinating, I think. And it's pretty intuitive, but it's really cool to see. So in red, you can see the prior non-controllers. Their virus came back. It came back quickly. They went up to high levels. This is expected. And they restarted ART pretty soon. But in blue, what I'm showing is that most of the prior controllers actually re-achieved control, which again, might not be surprising, but to actually see that happen in real time is fascinating. And now the study has associated with it a very robust tissue and blood biorepository where we're actually able to look at predictors and determinants of time to rebound, of set point, of achieving control or not. And so we're spending a lot of time on that. Another big focus of our program is, as I alluded to, immunotherapy to try to induce post-intervention control in individuals. And pretty much all models of control off of ART suggests that you need both a low reservoir state, so a small reservoir to begin with, and a really robust and sustained and not exhausted T cell response to the virus to achieve and then maintain long-term control. So I won't go through the schematic, but this is from a review we recently wrote, kind of outlining the various ways in which immunotherapy strategies can kind of induce this effect. But what I really want to spend time on is walking you through this experimental medicine clinical trial that we've done over the last 5 years, where we used a combination of different interventions that had each on their own shown promise in monkey models, suggesting that they could move the needle toward post-intervention control, at least in non-human primates. Steve designed this study maybe 10 years ago, and when I showed up at UCSF, he handed me the protocol and said, "How about you do this as your first project?" This was quite an intimidating study to carry out, and I think it sort of immunized me against all future studies, so nothing will ever be as complicated as this study. What we did in this study was we enrolled 10 people with HIV who had not been able to control their virus before they started ART. And we ran them through this gauntlet of immunotherapy. So there were 3 administrations of a conserved element HIV vaccine administered by electroporation. So what that means is that you administer the shot, and then you apply an electrical current to the muscle, just like electroporation sort of in the lab, to increase the sort of the uptake of the vaccine into the cells. It's quite dramatic. I would not care to do another study using that strategy again, but the participants really stuck it out. So we did that 3 times, 6 doses. Then we did a standard intramuscular MVA boost to improve the immune response against various elements of HIV. Then at week 24, we did a passive infusion of 2 broadly neutralizing antibodies against envelope and binding site. So these are IV infusions, basically monoclonal antibodies. Then we gave 9 weekly doses of a toll-like receptor agonist as part of a latency reversal / immune activation strategy to make the virus more visible to those BNAbs so that they would form immune complexes maybe and improve the immune response. And then we gave a final infusion of the 2 broadly neutralizing antibodies at week 34 and stopped people's HIV meds and watched them for up to a year to see what would happen. So this study opened on February 20th, 2020, which was a very bad time to open a complicated HIV cure immunotherapy study. It pretty much immediately shut down 2 weeks later because of COVID, and then we spent most of 2020 figuring out how to reopen it and actually do this in the midst of the pandemic, which is quite an experience. And just to give you a sense of the level of intensity, each of these 10 participants had about 60 visits over a 2-year period. So it's a 10-person study with over 600 visits. The results were, we think quite fascinating. So generally, if you interrupted ART in 10 individuals, you might expect, one to exhibit evidence of control afterwards. Even in most immunotherapy studies, it's been pretty much between 10 and 20% of participants. But in this study, 7 of the 10 participants actually exhibited really interesting post-rebound kinetics. So what I'll show on these graphs is basically everything synced up from the time that the virus reemerged. Everybody had variable time to rebound, but this is once the virus reemerged. So we had 3 individuals who had, what we would characterize as typical rebound. When the virus came back, it came back explosively and exponentially, and they went up to high levels and they had to go back on ART. We had, well, there's one person showing up here, but there are 2 lines on this graph, 2 individuals who had what we refer to as non-exponential growth. So really kind of sluggish rebound, where the virus came back, but it came back slowly over 6 to 10 months. And eventually they went back on ART, but this was not sort of the typical exponential rebound that we expect. What we think is most interesting is we had 4 individuals who exhibited what we refer to as durable partial control. And what I mean by that is when the virus came back, it kind of went up and then it came down and then it went up and it oscillated around a set point of 1,000 copies or less. And all of these individuals were able to maintain this level of control for 6 months or longer, and ended up restarting meds largely because of other things going on, like the need to get a COVID vaccine, or then the Mpox stuff happened in the midst of all of this as well. And then we had 1 individual who didn't rebound at all. He actually remained off of ART for 18 months, and eventually he and we all just sort of got exhausted by the level of monitoring that was necessary to do this in the context of research, and he elected to go back onto meds. So when you overlay the rebound curves from the non-controllers in this study who achieved post-intervention control, which is shown in green, what we expected was for them to look like the red lines in that observational study that I described before, where these are non-controllers, so their virus should come back and it should come back quickly. If you look, the green lines actually overlap mostly with the blue lines, which are the prior controllers from that observational study. And so we think that this post-intervention control phenotype is sort of recapitulated what some people are able to achieve spontaneously. So the question for that is why, how and why? And we're spending a lot of time and resources trying to figure that out. I'll just show a few things. So our Amelia Deichmann, our pharmacologist, has done modeling of the initial rebound phase, looking at that sort of intercept time point where the virus first comes back. So what you're looking at here in red are the non-controllers from the observational cohort. And as I described, the virus comes back with a high slope. In comparison, the prior controllers who re-achieve control have a reduced slope compared to the red. And then actually the post-intervention controllers in the study have an even more favorable slope than the spontaneous controllers in that observational study. And so we think that this represents the activity of the immune system interacting with the virus again and achieving through various mechanisms that control. Sorry, I don't know why this is so blurry, but most of the biological work that we're doing that has been really interesting has been in the T-cell responses. And Rachel in our group has done a really deep dive into this, and really the strongest correlate of achieving control in this small cohort is the proportion of cycling CD8 T cells as the virus is reemerging. And so you can see that the these proportions really shoot up in blue in the people who are able to then control their virus in comparison to the handful of participants in red who do not control their virus. And there's an indirect correlation, an indirect relationship between the proportion of cycling CD8s and what the set point turns out to be later on. There's no relationship with the reservoir size. There's no change in the reservoir size. And then there's no relationship with the BNAb pharmacokinetics. So most of these people have either washed out or the virus that's reemerging is resistant to the BNAbs. So we don't think that there's residual BNAb activity. So this has obviously been a big focus of our program over the last 5 years. We're now, trying to figure out how to expand on this. One of the things that we're working on this fall are trials that instead of doing the immunotherapy and then stopping meds, administering the immunotherapy at the time that meds are stopped, with the goal of really trying to intervene at the intercept when the virus and the immune system are re-encountering each other and drive people from a non-control phenotype to a control phenotype. So we have a number of studies that will be focused on that. We're also thinking about ways to improve the delivery of the broadly neutralizing antibodies. These are currently administered through an IV infusion and they wane over time. We're really interested in for example, vectored approaches using AAV vectors to actually get people to make their own versions of these broadly neutralizing antibodies so that they can be sustained. And then a big challenge with this study and a lot of the other cure studies that have been reported this year, including the Rio study that was done in the UK and the FRESH study that was done in South Africa, has been that when all of these studies were designed, we didn't know the time points at which the biology would be most interesting. Now we have a better sense of the timing of those things because we've done a few small trials in people. And so the groups that worked on all of those studies have banded together to form and design a new study that will be deliberately designed around collecting the right biospecimens to actually answer these questions. And so we're hoping that will get across the finish line at the NIH. So that's HIV. I'll shift now to COVID. So everything I mentioned in that section was planned or started before February of 2020. Those trials both launched in February of 2020. And then in March 2020, obviously the world shut down. So we had our first case of community spread in San Francisco on March 5th, 2020. And within a few days of that, our whole HIV program was put on hold because everything was put on hold. When that happened, I called up Steve and I said, what are we going to do? And he, was just basically supportive of us doing whatever we thought would make the most sense in response to the pandemic. And so we knew that there was going to be a lot of focus on understanding acute COVID and who was going to get sick and who was going to end up in the hospital. But not a lot of people were thinking about what would happen after people had COVID at that time. And we knew from our HIV program, which had been ongoing for 2 decades, that what we were actually good at was enrolling people, deeply phenotyping them, and following them for a really long time. And so we basically decided to apply that model to people without HIV who had COVID. We wrote the initial protocol over the next couple of weeks, and it was really focused around studying post-COVID immune responses. And actually, our initial funding was to collect biospecimens in part to provide to the National Biodefense Repository in the US to inform the development of vaccines. So when we opened in April of 2020, we were mostly encountering healthcare workers who got sick, taking care of people in the hospital. And you might remember that there were the cruise ships docked in the San Francisco Bay that had COVID outbreaks, and we enrolled a significant number of people who had kind of survived the very harrowing experience on those cruise ships. But probably within a couple of weeks of opening, we started to get outreach from people that were saying things like, I had COVID 6 weeks ago and it was a mild case, but cognitively, I, have all of these issues now, and I'm afraid, I'm a healthcare worker and I'm afraid to go back to work because I don't want to make a medical error, or I'm a surgeon and I'm worried about operating. Um, we heard from one person who wanted to participate who asked whether she could do the entire encounter lying flat because she had developed such bad dysautonomia symptoms that she had been unable to be, upright for a significant amount of time over the preceding 6 weeks. And a lot of these people were just normal people who had no prior medical history, and some of them were even people we knew. And so when this started happening, we thought this sure seems like it's going to be important. And we made a deliberate effort to make sure that our case report forms were basically capturing all of this potential post-acute symptomatology. About a week after we made these changes, this article came out in the New York Times describing, basically long COVID in people in Italy. And so that was quite striking to see. UCSF actually provided a lot of support for this effort early on. We actually had a grand rounds on long COVID in June of 2020. And our initial media coverage was from Australia. There was an ABC story on ADAPT and LIINC talking about long COVID and what was happening in Australia. So it was cool to be a part of that. Over the summer and fall of 2020, we basically became overrun with interest in the program among people with long COVID, and then with interest among their friends who were totally fine post-COVID, but wanted to kind of understand why other people were suffering. And so we quickly exceeded our capacity, and we also realized that our initial plan to follow people for 3 months wasn't going to work. And so we started following people basically for 2 years, and now we follow people indefinitely. By the end of 2020, the NIH had started to pay attention to this, and they actually convened a workshop on post-acute sequelae of COVID on Zoom, where everybody talked about what they were seeing or experiencing. And the result of that was, of all of that, was basically a massive congressional appropriation of over $1 billion in 2021 to try to study this problem, which is quite an unprecedented investment. And most of that money has gone into a program called Recover. So since April 2020 in LIINC, we've enrolled over 1,700 people. We've conducted over 8,000 visits. We've banked over 100,000 biospecimens, and we share these specimens really widely. We support a lot of collaborations. We do our own stuff, but we basically support anyone who's a good scientist who isn't doing something that directly overlaps with something that someone else is doing. We've also, the bulk of our program follows people with and without long COVID longitudinally, assessing everybody the same way, which I think is quite important. We also have an acute COVID program where we enroll people at the time of a first or new infection at this point, and then follow them prospectively to identify predictors or determinants of long COVID. We have a tissue biopsy program where we're doing gut biopsies, probably done about 140 gut biopsies by now. Lymph node FNAs, we've done maybe 50 of those. Lumbar punctures, we've done about 100 of those. Bone marrow biopsies, which I thought would be hard to enroll, but we've done about 30 of those. And we just started doing endometrial biopsies to look at changes in the female reproductive tract. And the goal of these procedures is to look at tissue-based immunology and also tissue-based virology. We have an imaging program where we do PET imaging using various sort of research tracers to look at immune system activity. And we're actually trying to tag now monoclonal antibodies to look at whether we can find residual virus. We have a clinical trials program that I'll talk about, and we've begun expanding to other infection-associated conditions, including ME/CFS, and we actually just started a post-Ebola program. And then since 2022, we've been part of the National Recover program. So many of our participants are co-enrolled in both programs. I'll just make a comment about these conditions in general. I think everybody here is probably familiar with them. But the comment I wanted to make is that actually, if you go back to the SARS-1 literature, it's really kind of uncanny. The scope of the SARS-1 pandemic was minuscule compared to SARS-CoV-2, but there are papers written between 2005 and 2010 that basically describe long COVID in people who survive SARS. And so, to me, that's really interesting proof of concept, and it's kind of eerie to read those papers. In the US, we are terrible at diagnosing and managing these conditions, but a lot of conversations with clinicians here in Australia over the last week, and actually I'm quite impressed by how much, I don't think things are great anywhere, but I think that they're better here than in the US. So that's made quite an impression on me while I've been here. I won't go into all of this, but this basically shows our conceptual framework for studying this problem. So we have kind of what we refer to as the upstream biological mechanisms that are things that we study in the research lab. So things like viral persistence or autoimmunity or mitochondrial dysfunction, et cetera. There's also what we refer to as downstream pathophysiology. These are things that we can measure in the clinic. So that's, organ system function, the function or dysfunction of various regulatory axes, dysautonomia, cognitive dysfunction. And this abnormal pathophysiology is what we at least think is really experienced by people as long COVID. So within LIINC, we have programs that are ongoing or in development, or in some cases wrapping up, studying each of these different biological and physiological mechanisms. The mechanism that we've really invested the most time on over the last few years has been SARS-CoV-2 persistence. And that's really because A, I think it's the lowest hanging fruit, and B, there's been funding available for that. And so this has been a priority in our research agenda. I initially did not really believe that this was a thing that was possible or that was real. So my framework was that, this was purely a transient virus. This review that came out 2 or 3 years ago cataloged all of the evidence that was emerging that went against that framework. And what struck me about this review was not really the results of any of these studies, because they're all small and often quite imperfect. But it was the fact that there were totally independent cohorts, totally independent research groups, totally different assays that were effectively coming up with the same conclusion through different measurements, that at least in a subset of people, components of this virus seemed able to stick around for longer than we thought. So when these data started to emerge, I basically said that I would only believe it if we saw it in LIINC, because I understood who was in the cohort. And so we partnered with David Walt at Harvard, who developed a single molecule array assay to measure SARS-CoV-2 proteins in blood during the post-acute phase. And we actually began by using samples that we collected before the pandemic from our HIV program, people with and without HIV from 2015, where it's impossible for those people to have had COVID, so that we could establish the specificity of the assay. So 2% of the pre-pandemic samples were positive. Those are false positives because it's impossible for them to be true positives, which means that the assay is 98% specific, which is pretty good. But when we looked at the post-COVID samples, and these are people regardless of whether they have long COVID, these are just people who had COVID, we saw significantly higher proportions with detected circulating antigens 3 to 6, 6 to 10, 10 to 14 months post-COVID. We've now extended this out actually beyond 2 years. And what I'm showing you here these are all samples that were collected before anybody on the planet received a vaccine, and when reinfections were basically still case reportable. So we don't think that this can be attributed to vaccination or reinfection. We've done a lot of work now to try to figure out whether this is actually associated with long COVID. And the answer is maybe. When we look at the WHO definition of long COVID, which captures a lot of different things, is very, a lot of heterogeneity, we don't see any signal. But it's only when we really try to deeply phenotype people by looking at the consistent presence of different types of symptoms that are actually affecting their lives that we see a signal emerge. So we think we're seeing relationships emerge between neurocognitive and cardiopulmonary symptoms and ongoing circulating antigen in the post-acute phase. But we don't really see a relationship with fatigue. And I think that that's because there's probably a lot more misclassification of a symptom like fatigue, where, a lot of people with post-COVID fatigue probably don't have long COVID. So anyway, these confidence intervals are wide. We are now expanding the study to try to narrow them a little bit. But I think that this will likely be confirmed and kind of get across the finish line. We also see indirect evidence of viral persistence. So this is a study that we've been doing with John Weary's group at Penn, where they look at the activation state of SARS-CoV-2 specific CD8 T cells. And what they find is about 30% of people in the post-acute phase between 3 and 12 months post-COVID have ongoing activation. Again, this is before vaccines. This is before reinfections. It seems to be more common among people with long COVID compared to people who have fully recovered. These responses also seem to be quite stable, which is interesting. And this proportion, about 30%, is actually what we see across multiple different types of assays, which is quite interesting. This is also consistent with a study we did with Nadia Rohn's group a couple of years ago using CyTOF. And so she identified, so the way we group people for this study is we basically looked at people who consistently met the long COVID definition over a year-long period compared to people who consistently did not meet the definition. So the gray area in between was sort of eliminated from this study. And what we found was increased expression of markers of exhaustion on SARS-CoV-2 specific CD8s and increased expression of markers of tissue trafficking on CD4 T cells. And so that suggested to us that there's potentially ongoing stimulation of immune cells and that it might be coming from the tissues. So we really think in LIINC that long COVID is a tissue-based disease. And so we've invested a lot in trying to understand that. We began by doing that non-invasively in our PET program. So what we do in that program is we take people post-COVID and we infuse a radio tracer that basically lights up activated T cells. And then we're able to look across sort of the head to toe, all of the different organs and tissues of interest to understand immune cell activity in those spaces. So the first thing that we found when we started doing this was actually that everybody post-COVID had more tissue T cell activation than scans that were done before 2020. So that is, concerning. We don't know what that means clinically, but we think that that means that there's sort of ongoing immunologic sequelae in kind of all of us from this virus. But then when we compare people with long COVID to people who fully recovered, we see increased activation in key tissue areas, including neurologic tissues like the spinal cord, vascular tissues, lymphoid tissues, gut tissue, and bone marrow. And those are all of the tissues that we are sampling in our tissue program now. And that's largely because of these findings. So these are data from our gut biopsy program, which is the first tissue that we went after. What we did in the study was we did flexible sigmoidoscopies with gut biopsies in people who had T cell activation in the gut in the imaging program. So these are people whose rectal wall sort of lit up in the imaging program. When we did, thanks. So when we did these procedures, we do RNA scope. Oh, wow. That's actually worse now. Um, and you can see here in green is SARS-CoV-2 spike encoding RNA in the lamina propria, so in the connective tissue layer. It's not in the epithelium. If we look at people during acute COVID who've had like a recent reinfection, we actually do see epithelial signal. But in long COVID, we see this deeper signal. And we're trying to profile the cells in or around where the signal is. They seem to be macrophages. And then in a subset of people, we can actually identify double-stranded RNA, which is shown in pink here, which should really only be present if the virus is actively moving through the life cycle. So that, may argue in favor that there's some, active viral replication. So we don't really know in what form this persistence is happening. Some people argue that it could just be fossilized RNA, just like junk that's doing nothing, or potentially inducing an immune response. Based on the fact that we see spike and occasionally nucleocapsid in blood, it could be that there's some sort of non-productive infection where proteins are being translated, but new target cells are not being infected. And then, the double-stranded RNA data suggests that there could be active replication, although we're hoping someone will replicate that. Recover's done a bunch of gut biopsies now, so we may see something coming out of that. So based on those observations, we launched our clinical trials program a few years ago. This is really mirrored after our experimental medicine trials in HIV. So our first trial was called Outsmart LC. I spent a lot of time thinking of acronyms. So this was monoclonal antibodies for long COVID. And what we did in the study was we enrolled 36 people with WHO-defined long COVID, did a whole battery of baseline measurements, then randomized them to get an infusion of an experimental monoclonal or placebo, and then followed them for a year. We did 1,000 different PROs. We did objective measurements of physical function, cognitive function, dysautonomia. We banked a lot of blood. And then a subset of participants were able to opt in to things like the PET imaging that I showed you. The overall result of the study was negative. It was quite disappointing, although I don't think really surprising. So there was no difference really in any of the primary or secondary outcomes between the treatment arm and the placebo arm at the primary endpoint, which was 3 months. There are a lot of reasons why that might be. I think the main reason is that there is not a clinically available test that we can use to screen people into a trial to know that the people have viral persistence. So that's a challenge for all of these studies. But there are a lot of other issues that we can go into in the Q&A if there's time. So this was, one of now many negative studies that have not shown a benefit in terms of clinical outcomes in long COVID. But what I want to sort of talk about is the exploratory work, which I think is kind of more interesting. So one of the questions that we're trying to answer is, did the monoclonals have any biological effect at all? And so from our tissue program, from our imaging program, it's actually quite interesting. There's like a pretty clear difference between the treatment arm and the placebo arm at 3 months in terms of T cell activation across multiple tissue regions of interest in the treatment group compared to the placebo group. These numbers are obviously small because it's a small study and a subset got the imaging, but I think it's quite provocative to suggest that there was a biological effect. We're doing all the proteomics work now, so we'll get more information. And then the other thing that we're trying to do is try to identify predictors of responsiveness. And I won't spend a lot of time on this, but basically what we think we've identified is that people are more likely to respond if they have low levels of their own antibodies to start with and have a higher area under the curve of the monoclonal antibodies. So low baseline immunity plus monoclonals sticking around and penetrating their tissues for a longer amount of time. So we're really trying to use this study to inform how to do the next study. And so that could be enrolling people who have lower levels of their own immunity at baseline. But I really think that what we need to do is a more aggressive dosing strategy and give people higher doses of antibodies up front and potentially dose them repeatedly, over a 3 to 6 month period to try to maximize the difference between the treatment and the placebo group. So we're actually working on a project now that hopefully will do that. So since that first clinical trial, we've done a number of other ones. We've just completed a study of the Shionogi antiviral and Citralvir to see whether we can see a biological effect. It's not developed around clinical outcomes. It's really a biomarker study. Next week, we're launching a study of an IL-15 agonist with the goal of basically targeting viral persistence, not by targeting the virus, but by improving the immune response. We're participating in a national study, NIH-funded study of baricitinib, which is a JAK-STAT inhibitor that has been studied a lot in HIV, is being studied in HIV-associated neurocognitive disorder. This will enroll 550 people at 15 centers across the US. And really, the primary outcome of the study is actually neurocognitive function on an objective test. We're participating in a TLR4 modulator study of this drug called bezisterim, which is more specific to a lot of the pathways that low-dose naltrexone is commonly prescribed for long COVID works on. This study is funded by the new US Department of War because the US military is very interested in long COVID because there's there are Marines with long COVID that can't, do their job. And then we are developing a combination study that would use Pemgarda, which is a modern monoclonal approved for prophylaxis in the US, plus antivirals. We're also developing a study called Prevent LC, which is really based on data from our group and other groups that have shown that literally the amount of virus in a person's nose during an acute infection predicts whether they have long COVID in 3 months. So probably dealing with that early on could make a difference. There have been basically no studies of preventing this untreatable condition. So I'm hoping that there will be investment in that. It seems obvious to do that. And then we're developing a biomarker program called Viper to basically address this issue that we don't know how to enroll people into clinical trials who might actually benefit. And it's really hard to get any traction with pharmaceutical companies because there's this inherent mistrust of how people feel. So everybody wants something that is measurable to get someone into a trial. Biomarker development for long COVID right now is total chaos. There are all these individual groups that have developed kind of their own assays on really small numbers of people, like a few dozen people, and claim to have the answer. There's been no head-to-head comparison. I have tried to do head-to-head comparisons, and there's never enough samples, and nobody can really ever describe who is in the cohort, et cetera. So this is a big problem. What we're trying to do in this program called Viper is basically take the phenotyping and participant-facing aspect of this out of the hands of the assay developers and instead say, we're going to develop a gold standard longitudinal cohort with tons of biospecimens, leukaphereses, gut biopsies, serial measurements, even within the same week within people that are highly, highly phenotyped, and distribute them in a blinded manner to all of the labs to generate their results, provide the results back, and then we can say, this assay seems to be the strongest. This is how to improve this other one. Here's how they compare head to head. Maybe we need a combination. And so we're hoping that this program will launch by the end of this year. And then the last thing, just to end, we're thinking a lot about long COVID, sort of beyond long COVID, and using long COVID as a model for other infection-associated conditions. There are very unique circumstances in which long COVID emerged. We all know this. Everyone was simultaneously at risk. Everyone was actively swabbing their noses at all times. Nothing else was circulating for 2 years. And people were broadly aware that bad stuff could happen after COVID. And so is really a unique circumstance that gives us our best shot probably ever to figure out one of these conditions. But I hope that, what we learn can then be applied to ME and chronic Lyme and post-Ebola, et cetera. And then obviously we want to be ready for, whatever happens to come next, hopefully not in our lifetimes. So in that vein, we're trying to launch at UCSF a Center for Post-Infectious Conditions and Syndromes, which we affectionately call Picasso. And that basically will kind of expand our long COVID paradigm to these other conditions and hopefully make us prepared for the future. So I'll end there. Thanks for your attention. Happy to take questions.
So, Mike, that was great. So on the HIV stuff, after all those immunizations and monoclonals, I mean, up to 20% of the CD8 cells proliferating. I mean, that's extraordinary. Were they people who had a low nadir? I mean, do you think they had a biased repertoire at that point to induce that? I mean, because you see that with CD4s in immune reconstitution disease.
Interesting.
That sort of really biased response. But
I don't think offhand there's any relationship with any of the clinical factors, including historical factors that we measured. The CD4 nadir, the reliability of what we have in terms of that information is often quite limited. People have like a ballpark of how low their T cells went, but medical care is so fragmented in the US that we often don't have the actual data. Rachel is really trying to understand. I mean, the proportions are, like you said, huge. She's really trying to understand what this is.
And then just a sort of comment about your monoclonal antibody study in long COVID. So we were involved in a bunch of the active 3, which was for acute COVID,
right?
But it was in hospitalized patients.
Yeah.
And all those monoclonals failed.
Yeah.
But there were correlations between sort of outcomes and those who had the lowest antibody when they got under the trial had a better response. I mean, so it's it's not an a priori, but it was across the studies, right? So just sort of interesting. And I think that's published now. So
the other thing that I think is very interesting about that observation is we know that there's a subset of people who seem to benefit from who have long COVID and then seem to benefit from getting a COVID vaccine. They feel that their long COVID has gotten better as sort of like therapeutic vaccination. And so one wonders whether it's a similar effect, that those are people who have, for some reason, lower responses that get boosted.
And just the outpatient trial of the protease inhibitor, the Shionogi protease inhibitor, did seem to give so they did follow people into chronic, and there did seem to be a lower rate of long COVID in the active arm of that. So, the idea about preventing this.
That was, so.
I mean, it's a bit soft, but yeah.
There was one study that was presented at CROI, I think 2 years ago, that showed a benefit. And then there was a second study that did not. So the data are mixed. But, the other thing is that it's not like, I mean, 5 days of treatment may not be during acute COVID may not be the answer. So there's data that suggests that it's not only the maximal viral load, but it's the duration of positivity or the presence of rebound that is associated with later long COVID. And so the trials that we're trying to develop are actually treating people early, but with more than just 5 days, and to basically treat them for the maximum that is allowable by the FDA, which is typically like 2 to 3 weeks, to really just try to shut everything down. I think that those are the types of studies that need to be done.
Very interesting talk. I have 2 questions about the second half of your talk. So the first one is, I was wondering if you can comment on the lack of relationship between the gastrointestinal symptoms and viral antigen persistence.
Yeah.
My second question is about the activated T cell phenotype that's localized in tissue, and whether that has any associate between the induction of, say, like localized autoimmunity.
I will start by saying I don't know the answer to the second question. We're doing all of the tissue work, the tissue immunology work right now. So a year from now, I will know the answer to that question. The first question, so actually I think it's a numbers issue. I actually don't think that the dot and confidence interval for the GI symptoms on that graph that you're referring to is super reliable, because generally really prolonged and debilitating GI symptoms in our cohort are pretty uncommon. So I suspect that there's not very many people represented in that specific result. Yeah, it's interesting because some of the initial tissue work in other cohorts for persistence were, for example, in people who had like pre-existing inflammatory bowel disease. In our study, it's all immunocompetent people who are not on immune modulators, and it's pretty rare for us to see people with debilitating lower GI symptoms. We have a lot of sort of motility issues on the area of like reduced motility. I think it'd be interesting to look in people with kind of pronounced lower GI symptoms like diarrhea. We haven't done that.
Can I quickly ask one?
Sure.
Question? Okay. In the first part of your talk, you were talking about the slow rebound versus like people who could sort of like bounce and sustain. And yeah, control HIV replication. So what differentiates these two groups? Like HLA, like, there was one group.
We have not found anything that differentiates the groups, but the numbers are so small, it's hard to really make valid comparisons. I mean, most people in that study were treated, had initiated ART within the first 6 months after their infection. But, it's a pretty homogeneous group of, mostly sort of early middle age Caucasian men who began treatment early on in their infection. There aren't a lot of phenotypic differences between them. We're trying to figure it out.
Cool, thank you.
Yeah.
Greg, and then we have Austin online.
Great talk, Mike. One question, one comment. And I think in the end, you talked about how there's this opportunity for sort of long COVID to tell us about other post-infectious syndromes and that sort of unique aspect of COVID coming forward and everyone being infection naive and then sort of following people. And I think we did with ADAPT, similar to LIINC, was a real opportunity. It's still relatively small numbers of people that we followed in those sort of early period. My concern, I suppose, is that now we're in a stage where it's so diverse, multiple infections, vaccination, the ability to characterize long COVID in a sort of clean sort of way is sort of reducing all the time. And that sort of leads in a little bit to maybe playing a bit of a devil's advocate around the viral persistence and how you exclude multiple infections and whether you have longitudinal samples with sequenceable virus to show that it's a homologous virus all the way through rather than multiple infections. How do you deal with that?
Yeah, it would be nice if there were an easy answer to deal with that. Yeah, I mean, I think that cohorts, like ADAPT and like LIINC that banked a lot of samples and collected a lot of data before things got complicated are really valuable. I appreciate the comment because I'm not sure that broadly all people, including the reviewers for our grants, realize how valuable those samples are. But you can't redo that now, right? I think our strategy has been trying to identify factors in that sort of that key phase of the pandemic where things were simpler, and then apply them to the modern era where things are complicated, but the numbers are so much greater. But I think we need both types of studies. And it's not even, I think that these studies, instead of just cross-validating findings between cohorts, actually needs to cross-validate things across eras, like the simple era and the more complicated era. Sequencing, I don't know. We're doing metagenomics now to try to address things like this, but I don't know whether we'll ever really be able to figure that out.
Maybe Picasso is appropriate for your Senate. It's pretty abstract, some of the concepts you're trying to get.
Okay, this question from Gail. So it's asking whether it's antigen persistence or immune dysregulation or
arrow B, arrow C, but we don't really know what direction the arrows go in. It's most intuitive to say like there is an acute infection, and then there's persistence of that infection that causes immune dysfunction. But it is equally possible that there is some immune dysfunction that is allowing us to detect persistence. And actually, we need to be targeting the immune system rather than what is persisting. And so I think we needed to do this first generation of clinical trials all targeting the virus to just show that it wasn't going to be an easy solution where, you know, people need 2 weeks of Paxlovid and then are magically cured. It was a lot harder to argue for giving someone a JAK inhibitor before we knew that 2 weeks of Paxlovid didn't work in everybody. Now I think we have a little bit more leeway to do things that are targeting the immune system rather than just the virus. And that has been, you know, even though people like to feel sad or, you know, complain about the negative studies that have happened so far, those negative studies, even though it would have been nice to get a positive result, actually really open up the opportunities for what can be pursued, you know, safely going forward. So I think that's a huge win from the first generation of trials.
Okay, and with that, I'd like to thank Mike for giving that.
Thanks so much.